So today we're going to look at protein synthesis. And protein synthesis is basically how our cells make proteins. And remember, everything about you is based on the types of proteins you make. Proteins are polymers made from ingredients called amino acids. There are only 20 amino acids in nature, but there are thousands of different types of proteins. So nature had to kind of get creative. Well, what makes all of our cells different are the proteins that they make. So when you were first created, you were a zygote, you were one cell, and you had this DNA inside of you. And then you went through mitosis in order to make new cells and become an embryo. Well, eventually those cells, which we call stem cells or undifferentiated cells, decided that they needed to figure out what it was that they were going to become. So this is what we call specialized cells. They now have their special jobs. Well, theoretically, your brain cells have the same DNA in them that your stomach cells have. But if you think of DNA as a cookbook, you know that cookbooks are divided up into different chapters. So depending on what you want to make, you go to a different chapter. Well, our DNA is the same way. So our brain cells use a different chapter than our stomach cells do. So therefore, our brain cells make different proteins. And those proteins say, hey, you're a brain cell. Then our stomach cells will make different proteins that say, hey, you're a stomach cell. So everything about your cells and what makes all of your cells different is the proteins that they make. But when I talk about DNA, I tell my students to remember the number 3. There's 3 letters in DNA. There's 3 differences between DNA and RNA. Every 3 letters on DNA is called a triplet, and that means something. Every 3 letters on messenger RNA is called a codon. Every 3 letters on transfer RNA is called an anticodon. There are 3 steps in making DNA replication. There are 3 steps in making RNA, and there's 3 steps in actually making the protein itself. So the number 3 is very, very, very important. Well, when we look at protein synthesis, there's actually 2 steps to protein synthesis. We have what we call transcription, and we have what we call translation. Now, this video is going to focus just on transcription. Transcription is how our cells make RNA in the first place. Now, there's actually 3 kinds of RNA. We have what's called messenger RNA, which is basically a photocopy of the recipe. If you think of your DNA as a cookbook, it is a cookbook that holds about 80,000 recipes. Now, we only use about 50,000 of those, but our DNA has the potential to hold 80,000 genes. That's like having a cookbook with 80,000 recipes in it. That's a huge cookbook. And most of the time, if you have a cookbook at home, very rarely do you use every single recipe in that cookbook. Well, our DNA is the same way. And our DNA is protected in the safety deposit box that we call the nucleus. Well, when our bodies need to make a protein, they don't need the entire cookbook. They just need one recipe from that cookbook. So what our bodies will do is our bodies will make a copy of the one recipe they need. And that copy is what we call messenger RNA, because that's carrying the message to the ribosome. The ribosome is where proteins are going to be made. So the messenger RNA is taking the message from the nucleus to the ribosome saying, hey, I need you to make this protein. Now, we don't want to be carrying around this cookbook, because if DNA gets mutated, we don't really have a way of fixing that. But if messenger RNA gets mutated, it's not a big deal because it's just a copy. If I tear that recipe or that recipe gets wet or that recipe gets smeared, I can just go and print off another copy of it. Or if I take a picture and that picture's blurry, I can just take another picture of that recipe. Well, our cells are the same way. Whenever we need to make a recipe, we will just make a copy of the one gene that we need. We don't need to be carrying around this DNA. We don't need to be carrying around all these 50,000 different genes. 50,000 different recipes. So our bodies will go and just make a copy of the one gene that we need. And this is a process that we call transcription. Now, all RNA is made the same way. Remember, we also have what's called transfer RNA. I call transfer RNA the measuring cups, because just like your measuring cups hold your ingredients for you, when we get ready to make a protein, the transfer RNAs are going to hold my ingredients. Remember, my ingredients are my amino acids. We also have ribosomal RNA, and ribosomal RNA is the blueprint that tells us how to make our ribosomes, and the nucleolus makes that for us. So transcription is how we actually are going to make the RNA in the first place, and this is going to occur in the nucleus. Translation is going to happen at the ribosome, and this is where the chef actually makes the recipe in the first place. So transcription is where we use DNA to make RNA. Now, messenger RNA is the most commonly made type of RNA we have in our body because messenger RNA can only be used one time. So once we make a recipe, we destroy that photocopy, okay? Because we don't want to leave it sitting around. If you think of messenger RNA as you've printed off one recipe, well, you're not going to leave that recipe sitting around. It could get torn, it could be crumpled up, it could get wet, it could smear. Well, these would be like mutations of that recipe. So since messenger RNA is fairly easy to make, when your body gets done with the messenger RNA, it destroys it. And if it needs to make that protein again, it just goes back and makes a new photocopy. And this is a process that occurs in the nucleus and is called transcription. And that's what we're going to focus on today. Now, just a few things to keep in mind. When we talk about DNA, we talk about this thing called the genetic code. And the genetic code is a sequence of nitrogen bases along the DNA. It's basically like reading a sentence. Okay? When you read a sentence, you get an idea of what's going on. Well, our bodies read these nitrogen bases. And the sequence determines what proteins are made. So number 1 here represents DNA. So you can see these nitrogen bases, and our bodies take these nitrogen bases and divide them into groups of 3. Okay? And so every 3 nitrogen bases on a piece of DNA is called a triplet. And that would be like a word in my sentence. Well, what that triplet actually tells me is one ingredient, and my ingredient is an amino acid. So GGA would be the code that tells me, hey, you need this amino acid. I need you to go get this ingredient. And then TAC would be another triplet. That's another code that says, hey, now you need to go get this ingredient. And then CTG, another triplet saying, okay, now you need to go get this ingredient. So when we look at DNA, we take it and we divide it into groups of 3s called triplets. And those triplets tell me what ingredients I need. Number 2 here represents messenger RNA. And notice messenger RNA is almost a mirror image of 1/2 of the DNA. Because when we print off a recipe, we can only print 1 page at a time. Well, when we make RNA, we only create 1 page at a time. And it's still divided into groups of 3s. But on messenger RNA, those groups of 3s are called codons. Okay? On DNA, it's called a triplet. On messenger RNA, it's called a codon. And yes, if you look, they're pretty much the same thing. But since they're in 2 different places, we give them 2 different names. And then this down here represents my transfer RNA. Transfer RNA are my measuring cups. They're going to carry my ingredients to the ribosome. Remember, my ingredients are amino acids. Well, they also have like little key teeth that are going to lock in place. And again, divided into groups of 3 letters. So every 3 letters on a piece of transfer RNA is called an anticodon. So up here, I have 3 letters on a piece of DNA, like GGA. That would be called a triplet. 3 letters on a piece of messenger RNA, which is what number 2 is, like CCU, that's called a codon. And then 3 letters on a piece of transfer RNA, like GGA, is called an anticodon. Well, we use these as sentences. Just like you have to read a recipe in order to know what ingredients you need, our bodies need to read the recipe to figure out how to make the protein. And remember, the ingredients that we make, or the ingredients that we use to make the proteins, are the amino acids. There are 20 different amino acids in nature. And so nature gets creative with how it puts things together. So for example, you want to make cookies. Well, you can go and ask all of your friends for different recipes that have cookies, chipped cookies. And so this recipe that you get from your grandmother might tell you to use dark chocolate chips. Then you have the same recipe from your best friend, but it tells you to use semi-sweet chocolate chips. And then you have another recipe from the person down the street that tells you to use white chocolate chips. Well, nature got creative. Even though the recipes are almost the same, just by changing 1 or 2 ingredients, you make something different. Well, that's how nature makes all these different proteins from just 20 different ingredients. Now, there are 20 different amino acids, there are 64 different types of transfer RNA. Because if you take those 4 letters, A, C, T, and U, and put them into groups of 3, there's 64 different ways I can do it. So for example, I could have AAA, I could have AAU, I could have AAC, I can have AAG. So that's 4 different measuring cups. And so therefore, since I have 64 different types of transfer RNA, some transfer RNA carry the same ingredients. And that's what this is talking about right here. This is an example of what's called a genetic code. It's actually very easy to read. We have figured out what the code, what the letters mean on a piece of DNA and a piece of messenger RNA. So just like you learn to read and you now know what CAT says and you now know what AND says, we now know what ingredients these letters are telling us. So of the 64 different types of transfer RNA, 61 of them are measuring cups that actually carry something. 3 of them are what we call stop codons. If you think of a recipe as a sentence, you know that a recipe starts or a sentence starts because it has a capital letter. Well, in our bodies, every recipe starts with the same 3 letters, and that's called TAC. So every recipe on our DNA starts with the exact same 3 letters. So therefore, every piece of messenger RNA starts with the same 3 letters. And therefore, every protein in our body starts with the same ingredient. Well, just like you know a sentence ends because you have a period at the end, we have 3 periods, or 3 possible periods, that we call the stop codons. Stop codons are basically empty transfer RNA. They don't have any ingredients. So when your ribosome, who's the chef, is reading the recipe, when it gets to a stop, it knows, okay, I'm done making the protein. Now, there's lots of different examples of genetic codes. This one's a little bit easy because you don't really have to figure out how to read it, but it shows you the 3 combinations. So let's say AUG is the very first codon we have on every piece of messenger RNA. Well, you find the A here on the side, you find the U up at the top, and then you find the G over here on this side, and you bring it all together. AUG is methanonine. That's the ingredient that those 3 letters tell you. So for example, if you had CCA, you would find CCA, there it is, and that's the ingredient called proline. Now, if you'll notice, like I mentioned, there's only 20 amino acids. So some of our transfer RNAs are pretty much all identical. So right here, using this one as an example, notice as long as you have CC as the first 2 letters, it doesn't matter what the last letter is. It's still all going to be the same amino acid. And remember, when you have a mutation is when I change these letters. So if I mutate CCU, and it becomes CCA, it's not a big deal. It's the same ingredient. But if my CCU becomes CGA, that's a big deal. Okay? That is the wrong ingredient. Because now CGA, if you'll notice, CGA is this ingredient called arginine. So I have the wrong ingredient, I'm going to make the wrong thing. That's what happens with mutations. Now, so you can have more than 1 codon for each amino acid. Like I was mentioning, this is just another chart that makes it a little bit easier for you to read. And the genetic code is universal, meaning it doesn't matter what creature you are. AUG will mean the same thing in every creature in nature. CCC will be the same thing for every creature in nature. Now, what actually happens in transcription? Like I mentioned, transcription is where we take DNA and we're going to photocopy it. And we use it to make RNA. We could be making transfer RNA. We could be making messenger RNA. The cell could be making ribosomal RNA. But messenger RNA is the more common because we make messenger RNA all the time. Like I said, anytime we need to make a protein, we have to go to our computer and we have to print off the recipe that we need. But every time we go to make a protein, we don't need to go buy new measuring cups. So transfer RNA is often recycled, but messenger RNA can only be used one time. So every time our cells need to make a protein, we have to make a new piece of messenger RNA. Now this is going to take place in the nucleus. And as you can see here, you can only make one piece of RNA at a time. Now, on a previous video, we talked about DNA replication. And DNA replication is how we make DNA. Well, DNA replication, you would have to photocopy every single page in that DNA, in that cookbook. Remember, DNA replication takes place before cell division. And we need to make sure that the children that these cells are making have identical copies of the recipes that they need in order to survive. So with DNA replication, we have to unzip and photocopy every single page in that cookbook. That would be like you pulling up a cookbook on the computer and hitting print and it printing every single page. Well, DNA replication has 3 steps. The first thing we have to do is, well, unwind the DNA and unzip it. It'd be like you have to open the cookbook. And it uses an enzyme called DNA helicase to do that. Then once you're open to page 1 and 2, you have to copy pages 1 and 2. And an enzyme called DNA polymerase does that. Then you'll turn the page and you'll copy pages 3 and 4. And you'll turn the pages and you'll copy pages 4 and 5 and so on and so forth. And then this third enzyme called DNA ligase will come behind it and glue everything back together so we have 2 nice, neat pieces of DNA. Well, guess what? RNA transcription has pretty much the same 3 steps of DNA replication. When we make DNA, like I mentioned, you have to photocopy every single recipe in order to make a new copy of the entire cookbook. Now, I know in modern society, most of you don't know, what do you mean by photocopy? You don't have Xerox machines. It would be like you pulling a cookbook out of your pantry or your cupboard and taking a picture of every single page in that cookbook and then printing it off and putting it in a notebook in order. That's what your cells have to do when you make DNA. But when we make RNA, I don't need every single recipe. I only need the one recipe, just one. I just want to make chocolate chip cookies, or I just want to cook sourdough bread. I don't need all the other recipes in the cookbook. So when we do RNA transcription, we will only need to photocopy just one page, just one recipe. Well, enzymes need to tell DNA what to do. DNA has to know, okay, what am I doing? Am I going to photocopy every single page, or do I only need one page? So RNA transcription is going to use different enzymes to kind of do the same thing. The enzymes have to tell DNA what to do. So we're still going to unwind DNA. Okay? However, we only need to open it up to the one recipe my cell needs to make. Remember when we made DNA, DNA helicase will attach and it will slowly work its way down your DNA. Okay? It will slowly unzip every single part of your DNA so you can photocopy every single part. Well, we don't need to do that now. So a different enzyme attaches, and this enzyme is called RNA polymerase. It's often called RNA polymerase 1. So when RNA polymerase 1 attaches, it doesn't have to attach at the top. It's going to go to like a table of contents. It's going to figure out where's that recipe that I need, and then it's going to open your cookbook up. It's just like you saying, okay, I need to go to page 20. So you open up your cookbook to page 20. You only need the recipe on page 20. Well, if DNA helicase attaches, that tells DNA we're going to photocopy everything. But RNA polymerase, often called RNA polymerase 1, will attach. And it says, okay, we only need to use page 20. RNA polymerase 1 lets you know, hey, I need to make messenger RNA. Right. Then we got to put it together. We have to copy it. So we often have what we call RNA polymerase 2. Some people say RNA polymerase 3. You just need to know it's a different enzyme. It's not the same enzyme. One RNA polymerase has just unzipped it, and the other RNA polymerase has to come and link everything together. So it's got to make sure that my A's link up with U's. It's got to make sure that my C's link up with G's. Okay. Now, in the case of DNA replication, DNA polymerase did that. DNA polymerase made sure that the A's and T's and G's and C's linked up. But remember, I'm using RNA. So I have to have a different nucleotide. I'm gonna have a different sugar. RNA is different than DNA. So I have to tell the cell you're making RNA. So that's why I have to use different enzymes. And then in the end, RNA ligase puts it all back together. Remember, to litigate or a ligament, lig means to glue. And RNA ligase will come in and make sure that my RNA is back together, and it will put my DNA back together. So, like you can see here, if this had enzymes on it, right here would be where like the first RNA polymerase is. It attached here and it just unzipped DNA to that point. So it found the start, it found the capital letter, and it followed its way down to the period, the stop. So it knows that that protein, it knows that that recipe stops right there. And then another enzyme will come in, RNA polymerase 2, and make sure that everything is matched up. My A's, my U's, my G's, my C's, all that stuff. And then RNA ligase will come in and make sure that my backbone is put together the way that it's supposed to. And it'll go back in after this RNA polymerase drifts off and put my DNA back together. So with transcription, transcription is how we make RNA. And this is going to happen in the nucleus. Now, like I said, the most common type is messenger RNA, and it will then leave the nucleus and go to the ribosome. So it's like you going to your grandmother's house and taking a picture of your favorite recipe. You don't need the whole cookbook. You just need the one recipe. So you go to your grandmother's house, you take a picture of that recipe, and then you take it back to your house so that you can make it. Because remember, the ribosomes are the kitchens here. The ribosomes are where the proteins are going to be made. So the cell has to get the photocopy of the recipe from the nucleus to the ribosome. So messenger RNA is allowed to leave the nucleus to go where it needs to go.